So let's go ahead and get started. And I'm going to attempt to do this on the PC. This is the first time I've used the PC version of whatever this app is. And one of the reasons why is I usually use my iPad, but they updated the iPad last night, and now the Explain Everything app, it cuts everything. It makes the screen really tiny. So I'm going to try to do this with the PC. And so to review, we went over the oxygen binding of hemoglobin versus myoglobin. And remember, the important thing here is I expect you to know the differences in the shapes and what the shapes indicate. So for example, here with myoglobin, what's that shape called? Pardon? It's hyperbolic. Okay, you should know what a hyperbolic curve looks like from calculus. And the shape versus hemoglobin is called sigmoidal because it's S-shaped. And anytime that you have this S-shaped type of binding or catalysis, what does that indicate? Allosteric. It's cooperative, okay? Now remember, allosteric can be either positive or negative, but here this clearly shows that it's positive because as the first oxygen binds, it makes it more likely that the second one, then the third one, and then finally the fourth one would bind. And some other things to point out, that at any pressure of oxygen, myoglobin is actually more efficient than hemoglobin at binding oxygen. And so therefore I wanted you to be able to explain the rationale of why we weren't myoglobin based and why we were hemoglobin based. We won't go in that whole discussion at this time, but you should know that. Well, are you not going to let me? And in this instance, the homotropic, usually those are activators. And that's where its own substrate or ligand will increase the likelihood of binding another substrate or ligand. You can have heterotropic, which can be both an activator or an inhibitor. And in that instance, for example, with the ATCase, it's inhibited by the binding of CTP. It's activated by the binding of ATP, and aspartate is actually its substrate. And then we've seen oxygen binding to hemoglobin as being an activator. Likewise, we also see where, we will see where 2,3-BPG, which is that bisphosphoglycerate, wherever it binds, it is an actual, it also affects, even though it is not an actual substrate per se for hemoglobin. This is where we left off, was the fact that there were these two models of allosteric enzymes and proteins. One is called the concerted method of binding, and the other one's called the sequential. And both of them rely on this idea of having a T form versus an R form. Okay, so as I mentioned in the last class, the R was for relaxed, versus the T is tight or taut or maybe tense. And R would bind the substrate or the ligand, if it's not an enzyme, it has a higher affinity for it. Whereas, I hate saying the word tightly since it's relaxed, but it makes it more likely for it to bind and easier for it to bind. Whereas the T has less affinity for the substrate or the ligand. Okay, so it tends to be the inactive form. There are some enzymes where they're both active, it's just that the T is less active than the R. Okay, usually for the concerted model, in the absence of its ligand, the protein is in its T form, okay? And then the presence of this will shift it from the T form to the R form, and I'll show what I mean by that in just a moment with a picture. And then changing from T to R, then the reason why this is concerted is the fact that it must change simultaneously. So all of the changes are concerted. So if you have a 2 subunit protein or enzyme, both sides have to change from T to R at the same time. It's a concerted action. Right, so what that means is, I really wish it doesn't do it. Nope. I was really hoping that there was like an advance. All right, And once again, this is just a hypothetical protein, and it has only 2 subunits, whereas hemoglobin, for example, would have 4. Let me move this over where you can see the entire thing. Okay, so here you can, whoa, all of us get sick then. Here you can imagine that, or see the fact that you've got this dimer, this dimeric protein. This is the T state. This is the R state. For whatever reason, the old book used, instead of K, we're used to seeing Ks, they used L. I don't know why. This is an equilibrium constant. So I will never use L on mine. It's going to be K. It's like an equilibrium constant. And so it should make sense that if it's an equilibrium constant, you have products over reactants, and it's just that ratio. If the ratio is high, then that means that it's going to prefer the T state at equilibrium over the R state. If it's low, then vice versa. Okay. Now, what happens is whenever a substrate binds, so as we see here, we have the substrate binds, and according to this model, it's going to bind more readily to the R state. And in doing so, what's going to happen? What do you suppose happens? It just thinks back to Lavoisier, to the Chatelier's principle in equilibrium. Oh, you guys can't see this if I cover up with my hand. And so I can't just cover up part of it. But what happens to the equilibrium when substrate binds? What does the Chatelier's principle state? What is equilibrium? Pardon?
When product formation and reactant information is equal?
Well, not necessarily. Well, I mean, the rate, yes. But now that won't affect here with respect to the R. But that is true. What else is there? What's another way of saying that instead of talking about the rates? What is equilibrium?
Like concentration.
The relative concentrations. And so what happens whenever substrate binds to one of the R's? What happens to the concentration of the free R? It goes down. So what must happen? It's going to shift to the left. So whenever what's going to happen here is, let me see if I can make it a different color. I don't know how to. Oh, don't. Thank you. This one. So what's going to happen here is, since it bound, it's going to shift the equilibrium, well, to the left, down in the way that they have it here. And so it's taking away at least, one of those, one of the members of the population that is the free RR. And so that's going to cause another one from the T side to come over to become R. And so that explains positive cooperativity. And so then if another one comes and binds, it's going to shift it down. So another T is going to have to come on over to take its place to re-equilibrate itself. Right, so I'm going to try to explain inhibition using a concerted method. Once again, and this is going to be really interesting. Let's go to just black. All right, so we have, and I'm not going to be able to do the really pretty colors, the shapes. So we have something that's RR at equilibrium with the T. And if it's concerted, it's going to be all or none. Hey, that's not too bad. I was afraid of trying to write on this. It was going to be really difficult. All right, keep it steady. So we've already seen, I'm going to call it an activator, which is an A. And so in comes the activator. Oh. I had a stylus. Actually, I just didn't get it in my bag, but it's actually kind of at a weird angle. And so I was really surprised that I could do but thank you. But yeah, I also have this little nubby stylus, which is completely worthless, but I got a real one. Okay, so we have an activator that comes in, and so I'm just going to do it over here. And whenever it binds, this is the same thing as what we saw before. Now we have R, but it's got an A attached to it. And so that's what's going to shift the equilibrium, just like what we've said before. So now the equilibrium is going to go this way. And that explains activation, cooperativity, positive cooperativity, or positive allostery. So what do you suppose is going to explain negative cooperativity or negative allostery? Since the A stabilizes the R, what will an inhibitor do?
Bind to the T.
It binds to the T. And so this is how the concerted method explains the inhibitor. So I'll try to do the ominous red. I can't think of faith integration for this one, sorry. Oh, whoops, it says R. It should be R, it should be I for inhibitor. Okay, and so it binds to the T. In doing so, it pulls the equilibrium towards the T side. And so it makes it less likely that, the reaction will occur if this is an enzyme. So that's how it inhibits. So this one's fairly simple. It's an all or none kind of thing with the concerted method. Are there any questions over this before we go to sequential?
So if an activator is used, it's all over push to deactivate.
Pardon?
If an activator is used, it's all going to push to R.
Correct. Well, it's for that you have to realize we're talking about an individual protein will all push to the R, and another one will come and take its place. Because that's the difference between equilibrium. We're talking about the big on the macro scale. So you think you have 100 protein complexes, and maybe it started off 50-50. Usually it's not going to be that perfect. And if you bind the A, it's going to pull one more over that direction. So then one of the T's will have to come on over to take its place. So now it's like one ARR, maybe 50 RRs and 49 TTs.
Okay.
So that's why it pulls it this way, that way, versus the inhibitor could pull it the other direction.
So is it the inhibitors or the activators that cause it from going from R to T?
Well, it helps to cause it to go to R to T. It may not be the only thing, right? Because there's going to be some equilibrium before. Maybe it's not 50-50. Maybe it's 70-30. Maybe the enzyme or the protein is 70% of all of them will already be in the R state. If you have a whole bunch of A's then added, think about this as a medication, like a stimulant. You take that stimulant, then what it does is it binds to the R's to pull them down to become ARRs, so more TTs become RRs, which becomes really difficult to say. You have to be very careful.
Yeah.
Very, very careful. So, but God knows my heart. So, faith integration. Hashtag. Okay, so no, my Twittering is more like Twitter for the dyslexics or whatever. I always forget to put hashtag first. Okay, so the sequential model is where the binding of one side affects the binding of the other side, but they don't happen simultaneously. That's where it's different than the concerted model. With the concerted model, the two subunits did it together, became RR or TT together. Here we can have if we just have two subunits, it can become RT. We're gonna have like a soundtrack on this one too.
Bioethics. Or not bioethics, business ethics.
And so, but yeah, so here you can imagine that we have, and I will attempt to, let me just do some generic color. So the possibilities would be on the far end, T, 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 R, and R, R. See, it's sequential. And you could imagine, then you throw in your inhibitors and you throw in your activators, and so you have an extra term or terms that are possible in this. Okay, so the change in the conformation on one side is induced by when the substrate or the ligand binds the enzyme or the protein. And then that affects the likelihood of the next one to bind. So similar to what we've seen with hemoglobin and talking about with hemoglobin in the past, we just didn't call it, oh, that's a sequential model, where the binding of the first oxygen helps the next subunit over go from the T state to the R state, to go from tense to relaxed. Okay, so this is what it's trying to show in this one here. Let me get it to where we can see all of it at once. Here we go. And so here it's a cooperative binding. So once again, they're only showing the substrate. But you can imagine now that the substrate could bind in 3 different populations, just like what they have shown here. It can bind, the initial binding on the T could force that one to go from T to R. That's the induced, fit. And then, since this one is an R, it can then cause the next subunit over to become an R. And by doing that, would make it, they're not showing it, but would make the likelihood of another substrate, that's really big, that's a fat arrow, but another substrate to be able to bind. Because if you notice before, it was the, Before, I hit the wrong one, it was this shape, so it wasn't capable of binding a substrate, whereas once it becomes induced, it can bind for the second substrate, the second ligand to bind there. Okay, and so that's similar to what we've seen and we've been discussing with hemoglobin. So the binding to 1 causes the next subunit over to have a higher affinity for the next substrate or ligand. Just like you can imagine adding in an inhibitor, and the binding of an inhibitor can force it to go this other direction. Well, I was going to get too far, but it can force it to go this other direction and can affect it to make it less likely to bind. And so, as you can see, it's going to be a little bit more complicated if we try to do the R's and the T's, because you could have, additional things coming off. We could have an inhibitor binding here or here. You could have, the substrates or the activators binding to go toward that direction. So I'm not at this point going to attempt to draw all those in on here. Instead, I'll let them do it. And so here we've got the 2, what it would look like with respect to something that's got a tetramer, similar to hemoglobin. There we go. And so if it's concerted, there's only those 2 populations that exist. It's either all going to be, I can't remember now if this is going to be, the R state, or, I'll say that this one's the R and this one's the T. Doesn't really matter so much. But you can see that the binding of 1 will help make it more likely to bind 2, and so on and so forth. But we can have it still in either the R state or the T state. Whereas if it's sequential, we have much more complicated. And we haven't even started adding in activators or inhibitors. So as we mentioned before, BPG, if you're doing hemoglobin, could actually add another dimension, either coming up out of the board or going, and then if you have an inhibitor, it's going to go away from the board. And so it becomes multi-dimensional. But it looks cool, but it's a little hard to wrap your head around. But it's going to affect the dynamics here. Are there any questions on concerted versus the sequential? Concerted, all subunits change at the same time. Sequential, they change one at a time. You just have to remember that the inhibitors pull it towards the tense state, activators pull it towards the relaxed state. So now let's talk about the oxygen binding to hemoglobin and the medical implications here, and actually how this affects blood pH. And this is actually to show you the buffer system. This is the buffer system that our body uses in the blood in order to maintain the pH. So it's called the Bohr effect. And so what you see in this instance is we have oxygenated hemoglobin. That's what this HbO2 is to indicate. This is where oxygen has bound to the hemoglobin. Technically, it's coordinated to the iron. The iron's in the heme's attached with free protons, to where if the hemoglobin becomes protonated, then the oxygen is released. Okay, so if we think about it that way,
That's interesting.
that's good. Perhaps you want to expound upon the
Well, like the tissues are, I thought usually slightly basic, but that doesn't make sense for oxygen would be released.
Well, it depends on what the pKa is. Remember, they would just have to be more, it has to be more acidic than the pKa of the hemoglobin that's bound, that binds it, not pH 7.
Sure.
Okay, so that's where some people get really confused. They forget and they think, oh, it's got to be below pH 7 or above pH 7. No, we're talking about the pKa's here of hemoglobin. However, what is true, let's say that you've been working out, you've got your muscle. What happens to the pH in the muscle?
Doesn't it go up? Or down? Down.
It goes down. And so why would that make sense? Because what's the muscle going to be needing? It's going to be needing more oxygen. So that way the hemoglobin that is there, or the myoglobin for that matter, of course myoglobin members all or none, the hemoglobin that's there is going to be able to release the oxygen that it has. Likewise, the same thing is going to, so then we talk about, the lungs. Relatively speaking, do you think the lungs are going to be more acidic or more basic than the pKa of the hemoglobin? Basic, because you want the oxygen to bind in the lungs. And then it goes to the tissues in the brain where it's below the pKa of the hemoglobin. Okay. Carbon dioxide also promotes release because, and this is the reason why carbon dioxide does, and it's the buffer that's used. Of course, carbon dioxide is a gas. It is not polar, even though there's a big, if you think, just to do a review since it's been a while for organic chemistry, but the Lewis structure for, it helps if I draw. The Lewis structure for carbon dioxide looks like this. It's linear, and so the two dipoles cancel each other out. So it's nonpolar. And because of that, it's not very water soluble. That's the reason why, I should have waited to open my Diet Dew right there. But it's the reason why when you first open your soda and it goes psh, because the carbon dioxide is going to escape. Okay, and so what happens is within the body, we're going to be able to have it react with water to form, using the enzyme carbonic anhydrase, to form carbonic acid. And of course, carbonic acid is an acid. It gives up, become bicarbonate and the hydrogen. So by the binding of carbon dioxide, or having the presence of carbon dioxide here is going to release the proton. Then if you go up here, then the proton is going to be able to affect the ability of the oxygen to be released, which should also make sense once we get to the muscles, which would have higher concentrations of carbon dioxide, lower concentrations of oxygen, so on and so forth. So this is one way that we have to monitor the pH of our, of the blood, as well as be able to utilize oxygen and get rid of carbon dioxide, which, by the way, just in case you're going to forget this come next semester, but that way I can say, at least I said it once, carbon dioxide is as oxidized a carbon you can get. So if you think about your fats being a major energy source, especially saturated fats, those are highly, highly reduced. They're just carbons and hydrogens, and at the very end they have an oxygen, two oxygens. So what happens though is our body burns up that fat completely and turns it all into carbon dioxide that is as oxidized as it gets. So we have gotten all of the energy that we can out of that fat, or that sugar. But sugars are already more oxidized than fat since they all have those alcohols on them. So, yeah.
Is that why, like with diabetes, like their blood is more visibly acidic because more sugars are tending to bind to their red blood cells, so they need more oxygen?
So, did I just hear beeping? Okay. Yes, that does affect affect it because it's the ability of glucose. Glucose will mess lots of things up, as we will talk about next semester. So it binds to lots of different things and can affect it. Okay, because of the antigens and everything that's expressed in the cells. It also affects transportations. It affects other metabolic processes and cell signaling. That's why you can have such a wide array of effects from, eyes to just not being able to fend off, bacterial infections and things like that.
So this is the reason that we release lactic acid when we work out, like is it recruiting oxygen?
No, what we're going to find out next semester is that what happens is when you first get through your oxygen stores in your muscles to where you've done as much of the aerobic oxidation that you can really quickly, it will then convert to going to take the pyruvate down to lactate. And then, but that does help because then the lactate itself is lactic acid, but it looks like lactate. So it's giving off the protons, so that way when the hemoglobin or myoglobin gets there, it's going to release the oxygen more readily. So kind of a catch-22 or circular logic in some ways, but yes. But no, it's not directly.
Where you get like most ATP.
No. Pyruvate is the end product of glycolysis. Get it on the record. This is just a little cartoony form of saying the exact same thing. So we can see that, once again, we have, whoops, you can't hardly see it. Let me make it smaller. Okay, so we have oxygenated hemoglobin. It gets to the muscles where the muscles have a higher concentration of carbon dioxide, and if you've been working out, the lactic acid will be giving off the protons, so it allows for the oxygen to be released by the hemoglobin, the binding of the carbon dioxide, and it's protonated. When it gets back to the lungs will have a different pH, and so since it's going to be above the pKa there, the proton gets released, the carbon dioxide gets released, and the oxygen comes, and it's repeated over and over again. But that's called the Bohr effect. So there's a little less complicated way, and then the preceding slide is the one that shows you step by step of how carbon dioxide does directly link there to the actually working with water to affect the pH of your body.